I'm pleased to introduce our next speaker, Dr. Danika Bannasch. Dr. Bannasch is a veterinary geneticist who specializes in canine genetics. She obtained her Ph.D. in molecular biology from Princeton University and her DVM from the University of California at Davis. She has been a professor at the School of Veterinary Medicine at the University of California, Davis since 2001. She was appointed the Maxine Adler Endowed Chair in Genetics in 2013. In 2019, Dr. Bannasch was awarded the International Canine Health Award for her research accomplishments in canine genetics. Dr. Bannasch has published over 100 peer-reviewed manuscripts, and her laboratory has identified the causative variant for 16 inherited diseases in dogs and horses, and collaborated on the identification of many more. Her research goals are to improve the lives of dogs through understanding the relationship between their genetic variation and the resulting phenotypes. In her spare time, Dr. Bannasch breeds dogs and competes in conformation, agility, hunt tests, tracking, and barn hunt with her Nova Scotia duck-tolling retrievers and Danish-Swedish farm dogs. Please post any questions you have in the chat box during the presentation, and Dr. Bannasch will answer as many as possible during the live Q&A. Welcome, Dr. Danika Bannasch.
Thank you for that introduction. Today I'm going to talk to you about chondrodystrophy, what it is, and why we seem to like it so much. So first we're going to start off with a definition. So chondro means related to cartilage or bone, and dystrophy stands for tissue degeneration. So that name was actually coined in the mid-1900s by Hansen, and the reason why he was interested in studying chondrodystrophy and why he named it is because in the late 1800s and early 1900s, veterinarians had recognized that some breeds of dogs, in particular lovely dachshunds shown here, were predisposed to acute paralysis or sudden paralysis, and they wanted to figure out why some of these breeds kept coming up with this sudden paralysis. So Hansen did a really complete assessment of the intervertebral discs and the long bones in a set of breeds, the Dachshund, the Doxabraki, Pekingese, Spaniels, and French Bulldogs, and normal breeds. And he identified that they had intervertebral discs that degenerated after birth, in addition to some defects in the growth of the long bones of the legs. And so he named that disorder chondrodystrophy. He also defined the clinical disease of intervertebral disc disease and designated the type of disc herniation in these dogs as type 1. Later in the 1970s, Braun did the similar types of studies with beagles, and so I've put the little picture of a beagle at the top because that breed has also been demonstrated to be chondrodystrophic. Now, in veterinary medicine, there's been sort of gross misuse of this term over the years because not everyone agrees about which breeds are chondrodystrophic. Even when I went to vet school, there were sort of different breeds kind of lumped in there, and then years later, I would learn about a different set of breeds. And if you look in publications, everyone seems to have their own set of breeds that they think are chondrodystrophic. So there's no wonder it's a little bit confusing because even the experts are a little bit confused. But really, these are the only breeds that were demonstrated to have chondrodystrophy, and we can maybe now add one more to that. So a lot of this has been resolved because we found the variant or mutation that's responsible for this chondrodystrophy phenotype, and I'm going to tell you a little bit about that. I have to tell you that the way that we found it was kind of by accident, and I guess a lot of science progresses that way. So we were studying a breed of dog called the Nova Scotia duck-tolling retriever, which is the smallest of the retrievers, and they range in height from 17 to 21 inches at the shoulders. And what we noticed is that there were some really short tollers, there were medium tollers, and there were tall tollers. And some of the short tollers had very crooked front legs. So we called this disease skeletal dysplasia, which means that there's something wrong with the skeleton. Very simple term there and broad term. And the part where the front legs are crooked is called valgus. That's the veterinary term for it. And these dogs also have some curvature of their radius. So in order to study this further, we took radiographs or x-rays of the dogs, and that's shown on this slide. And what we found is that there was a really big range. So these are radiographs of the forelimb. So the head is facing to the right and the rear is facing to the left. Hopefully my right and left is the same as your right and left. And you can see that there's quite a bit of variability. And rather than trying to quantify that variability, because we weren't really sure how to categorize the dogs in the middle, we just used the extremes. So we used the really severely affected and the really straight-legged dogs and performed something called a genome-wide association. So this was a way for us to try and identify what region of the dog genome had the mutation that conferred this phenotype. So this is called a Manhattan plot, because you can see it kind of looks like a Manhattan skyline there. And what you always want to see in one of these plots is dots that go above the red line. So in this case, we had a really significant association on canine chromosome 12. So this is for skeletal dysplasia and Nova Scotia duck-tolling retrievers. I promise I'll get to chondrodystrophy soon. If we look at specifically just chromosome 12, these are those same dots, which are markers along the chromosome. And the little red bar, or the big red bar, shows the area where all of the affected tollers had identical copies of the chromosome. So they were homozygous for the same region of chromosome. And that is a huge region. So it contained over 40 genes. And we looked through there a lot, and we didn't find any obvious mutations, and we were a little bit stumped. And we couldn't quite come up with a great idea to narrow it down. So what we decided to do is to use other breeds of dogs. So, of course, you know that dogs come in all sorts of shapes and sizes, and there are lots of little breeds and big breeds, and we thought, well, maybe some of these other breeds would have something in common with the Nova Scotia duck-tolling retrievers that were affected with skeletal dysplasia. So what we found is shown on this slide. So this is a graph that shows whether or not there's variability across the chromosome. And what we found in a tiny region within that red region that I showed you for the skeletal dysplasia tollers is that there were certain breeds of dogs that all had the exact same DNA sequence as the tollers, and they also had the two copies were identical, so they were homozygous. So this told us that these breeds shared this region, and what was really striking is that they were the chondrodystrophic breeds that had been defined by Hansen. So we thought, wow, okay, maybe the skeletal dysplasia is chondrodystrophy. So we did a few more association studies. So this is the first association study I told you for skeletal dysplasia and tollers. And that showed us that there was a region on chromosome 12 that was associated. And then we compared chondrodystrophic breeds to all other breeds, got a similar association in the same region. And then we actually looked at breeds or dogs that had had Hansen type 1 disc disease, which again was one of the sort of defining features of chondrodystrophy, and got the same region. So that was really great, really excited about that. And we looked in the literature to see if anyone else had ever identified this region. And we found two studies that had. One was the fine mapping of a locus controlling leg morphology. And this was a study done by Elaine Ostrander's group in Portuguese water dogs, where they were looking at a variant that affected leg length, and it was actually to the exact same region. And another study done by Merritt Fredholm's group that showed that in standard dachshunds, so the larger size toxins, that there was a major locus that affected disc calcification that also mapped to the same region. So that was really exciting for us, and we thought, okay, this is great. This really is the chondrodystrophy region. But neither of these groups had identified the actual mutation, and we started looking ourselves to try and figure out what that was. So this is a sort of a picture of the genes across this region, and we sequenced all the genes. We didn't find any mutations. The previous groups hadn't found any mutations, and so we started doing something a little bit crazy, which is what we call visual inspection, where we actually just looked at the sequence across this region. So this is a pretty big region, and it took a long time to look at it. But what we found was that there was a really weird sequence anomaly. So this occurred in dachshunds with intervertebral disc disease or intervertebral disc herniation. Skeletal dysplasia affected Toller, but not in a Saluki, which was a normal-legged dog. And these sequence reads are highlighted in pink. And what they told us is that there was an insertion of a new gene in this area, and the new gene is called FGF4. So we'll go to the next slide. So normally, FGF4 lives on canine chromosome 18, but what happened in chondrodystrophy is that a copy of the FGF4 gene got inserted on canine chromosome 12, right in this region where all the chondrodystrophic dogs are homozygous and all the skeletal dysplasia tollers are homozygous. So when we first found this, we were really excited because it's not the first time that this gene was identified to jump. So it seems to be kind of a jumping gene. So in 2009, Elaine Ostrander's group had identified that there was a copy of FGF4 that was associated with chondrodysplasia in dogs. So chondrodysplasia is a term that they used to define dog breeds that had short legs based on their breed standard. And so these were sort of more extreme short legs than the tollers that I showed you. So once we identified that it was the same gene that had jumped on chromosome 12, that made a lot of sense because these two disorders are sort of related to each other, they're similar, but one thing that we knew is that the chondrodysplasia dogs didn't fully explain disc herniation because there were dogs that had chondrodysplasia that weren't predisposed to disc herniation. So now it sort of all made sense that there was a second place that this gene had jumped, and that second place was causing a kind of different set of problems. But they're both caused by the same gene that inserted in two different places. So this is a little bit different than normal mutations that we talk about where there's a normal copy of a gene and then an abnormal copy. In this case, the normal version of the chromosome doesn't have this extra gene inserted there, and the abnormal version of the chromosome has an extra gene. I put some names up here because both by my group and other groups over the years, we seem to keep changing what we want to call this. So it can be called the chromosome 12 retrogene, or CDDY, which is shortening for chondrodystrophy, or FGF4-like 2. And the same way chondrodysplasia can be called FGF4 retrogene on chromosome 18, chondrodysplasia, or FGF4-like 1. So when there's a copy of a gene that gets moved to a new location, the name for that is a retrogene. And so you might hear this referred to as the FGF4 retrogenes. Okay, so let's look at what FGF4 does normally. So FGF4 stands for fibroblast growth factor 4, and these are some mouse embryos, and where FGF4 is normally expressed is stained in purple or pink. So in the image on the left, you can see this staining that's actually where the external ear develops early in embryogenesis. And then the image on the right, they're staining both along the somites, which eventually will become a spinal cord, and the little region at the tip of the developing limb buds. And so it was shown in mouse experiments that FGF4 is essential for the limbs to develop normally. And this gene is normally completely off in adults. So it's only on during development. So what we suspect is happening is that the retrogene, the extra copies of FGF4, are driving higher expression of FGF4 during embryonic development, and that leads to the phenotypes that we see in dogs. So we were not able to study dog embryos, but we were able to get some RNA from intervertebral discs from puppies that both had chondrodystrophy and didn't have chondrodystrophy, and showed that FGF4 expression was really, really high in dogs with chondrodystrophy in the intervertebral disc when it should have been basically off. So we think that the reason why we see this phenotype is because FGF4 is expressed at the wrong time and at really high levels where it shouldn't be. So let's look at the overlap between these two FGF4 retrogenes. So there's a list of breeds here on the left and a frequency of how often we find the FGF4 retrogene on chromosome 12, so chondrodystrophy, and the FGF4 retrogene on chromosome 18. What we see is that the breeds, beagles, dachshunds, French bulldogs, and spaniels have a really, really high allele frequency, so they're basically homozygous for the mutation. It's probably why Hansen kind of figured out that there was something a little bit odd about them. But they don't, some of those don't even have chondrodysplasia at all. So the set of breeds that Hansen defined as chondrodystrophic just have the FGF4 retrogene on chromosome 12, or chondrodystrophy. And there's a set of terrier breeds that have short legs, and they're not predisposed to disc disease, and they just have the retrogene on chromosome 18. And then there's some breeds that have both, and those are the breeds that we think of that are particularly short-legged. So it seems like when you have both retrogenes, the dog's legs are even shorter. So let's talk a little bit about the intervertebral discs that I've been talking about. So this is sort of a picture of a normal canine spine showing the vertebral bodies that are connected that make up the back. And you can see the spinal cord that runs inside those vertebral bodies. So we want to protect our spinal cord because it takes all of the nerves from the brain all the way down the back and throughout the body. And the intervertebral discs are these wonderful little soft cushions between each of the vertebral bodies, and they help cushion and support the vertebral bodies and also protect the spinal cord a little bit. So the intervertebral discs are made up of two structures. One is the nucleus pulposus, and that's this sort of spongy part in the middle. And the outer ring around that is called the annulus fibrosus, and that's a bit of a fibrotic ring that contains the nucleus pulposus. So if we look at a dog's skeleton, there are 27 discs along the body, not on the tail, which can have a number of discs. And all of those discs are, intervertebral discs, are meant, like I said, to support the movement of the spine. And let's look at this. So here's a dog running, showing you how that back can torque, and actually that's that spot where you see the most movement on the dog, right behind the rib cage, is one of the main locations where we see disc herniation. But there's also movement left and right that happens with these discs, and we know lots of dogs that love to curl up into a little tiny ball, so you can imagine how flexible this structure needs to be. So we said that chondrodystrophic dogs have degeneration, so let's look at that more closely. So this is a cross-section through a spine of a dog that has a normal disc. It's non-chondrodystrophic, and you can see this glistening blue structure in the middle. That's a normal nucleus pulposus that's acting as a cushion, and it's going to cushion those vertebral bodies so that they don't run into each other. And this is the same image of a 2-year-old dog that has chondrodystrophy, and you can see that that nice gelatinous cushion is gone, and what it's been replaced by is this sort of cartilage that is a lot harder and less cushiony. There's a loss of water content from it, and there's also a loss of flexibility in the chondrodystrophic dogs. So if we look at this at the cellular level, which is what Hansen did, we can see that the cellular structure is also abnormal. So in this work that was done by Dr. Brian Murphy at UC Davis, he showed that the nucleus pulposus in Nova Scotia duck-toling retrievers that have chondrodystrophy is abnormal. So these are actually the slides of normal puppies. So these are dogs, these are Nova Scotia duck-toling retrievers that don't have chondrodystrophy. And that white area is an area of liquid and special substances that give it this sort of gel-like structure. And this is what the chondrodystrophy puppies look like. So this gel-like structure has been replaced with cartilage, and it's solid. So it kind of goes along with those images I just showed you. What we showed in the Nova Scotia duck-toling retriever is that the discs are replaced by cartilage as early as 10 weeks of age. So they're still degenerating throughout life, which is makes it appropriate to call this disease dystrophy, but it seems to be happening even earlier than Hansen had originally thought. So how do we get from here to disc herniation? So types of disc herniation, the one that is common in chondrodystrophic dogs is called Hansen type 1. And this is where this hard nucleus pulposus actually breaks through the annulus fibrosus, and it goes upward. And I showed you in the previous slide that what sits here is the spinal cord. So if this disc breaks through quickly, rapidly, it can hit the spinal cord, it can cause compression of the spinal cord, it can cause inflammation and bleeding, which can compress the spinal cord, and this leads to severe pain, and it can lead to paralysis. So if a dog herniates a disc in their neck, then it can cause paralysis in all 4 limbs, and if they herniate discs in the back, it can cause paralysis of just the hind limbs. Okay, so let's just revisit some terminology for a second. So it seems that the discovery of the chondrodystrophy mutation has led to an increased interest in these terms by people. So IVDD stands for intervertebral disc disease or degeneration, and that's really a broad term that encompasses dogs with degeneration, like I showed you just at the cellular level, up to dogs that have herniated a disc but are subclinical for it, so they don't show any clinical signs associated with the herniated disc, all the way up to dogs that have intervertebral disc herniation. So I think when people see the results of testing and they see IVDD, they only think that that means herniation, but that's not necessarily the case. Because we know about the cellular structure that has changed in these dogs from early on, they still have disc disease. They just might not have become paralyzed from it. So we wanted to study more about how this mutation and the FGF4 retrogenes contribute to disc herniation. And we did this in a study using cases of dogs that had had disc herniation, were seen at our teaching hospital, and had had surgery as a result of the disc herniation. And we evaluated those dogs for the age of surgery, whether they had radiographically visible disc calcification, their weight, their breed, and then we genotyped them for both chondrodystrophy and chondrodysplasia. We actually did this just from a biobank that we had pre-existing in the hospital. So to give you an idea how common this is, just by random, when we generated a biobank, about 10% of the samples were from disc herniations. And that's because, at least in our hospital, we see a lot of them. We treat one case of chondrodystrophy caused disc herniation a day. So about 360 cases a year. So what we found from this analysis is that the FGF4 retrogene on chromosome 12, or chondrodystrophy, acts in a dominant fashion to decrease the age of onset of disc herniation by 2 to 2.5 years. So dogs with one copy of chondrodystrophy are 2 to 2.5 years younger than dogs that have disc herniation without chondrodystrophy. And I maybe didn't mention that before, but there are other ways that a dog can herniate a disc, just like there's lots of ways that people can have back problems and disc problems. But what chondrodystrophy does is it makes it happen earlier. It also acts in an additive. So let me back up. Dominant means you only need one copy to see the effect. Additive means that 2 copies gives you a worse effect than 1 copy. So on disc calcification, chondrodystrophy is additive. So 1 copy increases the risk of disc calcification, and 2 copies increases the risk even higher. Also, the rate of having at least 1 calcified disc differed significantly by breed. So it was highest in dachshunds, and then French bulldogs, and then mixed breed dogs, and then other purebred dogs. So there's definitely something else affecting the frequency of disc calcification in dogs. I also want to say something about dominant inheritance here. Remember that I told you that the reason why the FGF4 retrogene causes problems is because there's overexpression. There's more FGF4 protein at the wrong time in development. And that happens with just 1 copy of the mutation, right? So if there's 1 copy of FGF4 inserted on chromosome 12, that still leads to overexpression. So that's why you see a phenotype with just 1 copy. All right. So when we looked in mixed breed dogs to try and get away from the breed effect, since there's selection for chondrodystrophy and chondrodysplasia, we identified that only chondrodystrophy was had a significant effect on disc herniation, that chondrodysplasia didn't seem to be contributing to it. So that was important because we wondered why dachshunds have the highest frequency of disc herniation, and we thought maybe it was because they had both copies of the FGF4 retrogene, but it doesn't seem like that's the case. We also looked at the relative risk of disc herniation in breeds that segregate chondrodystrophy. So these are breeds that are not all homozygous for the mutation. Some of them have a normal allele. And we found that the relative risk varied from 5 to 15. And what that means is that dogs that have chondrodystrophy are 5 to 15 times as likely to herniate as dogs that don't have it, or more likely to herniate than dogs that don't have it. So that tells you that they have a higher risk. People always want to know what percentage of dogs with chondrodystrophy will herniate, which is actually a really hard question to answer. But I found one study that did a pretty good job of evaluating that. And that was a study looking at clinical disorders observed in a beagle breeding colony. So these were laboratory beagles raised under identical conditions, but they were treated pretty well. They had active group play sessions, and they weren't just in small cages or anything. They were laboratory dogs, but they had really good happy lives with lots of interaction and playtime. And since beagles are homozygous for chondrodystrophy and they don't have chondrodysplasia, we could tell that this was just limited to chondrodystrophy. And over their lifetimes, 10% of them had disc herniations. So this is where we have used this information to say, well, if the dog has a copy of chondrodystrophy, it's about 5% likely to herniate. That being said, estimates in dachshunds have been as high as 25%. So this also might differ by breed and other things that are going on in the breed. So the next thing we did was we investigated the effects of chondrodystrophy in tollers specifically by taking a closer look at their discs. So we performed whole body CT scans on Nova Scotia duck tolling retrievers, and this means that we looked at them really, really carefully with a special CT machine that's really good at evaluating bones and mineralized discs. So you can see here in these pictures, there's little white specks between the vertebral bodies on each of these pictures, and those are areas where the discs have mineralized. So we're doing a lot with this study with the CT scans, but some of the first data that we have back is shown here. We evaluated 22 Nova Scotia duck tolling retrievers, and in the 3 different genotype classes. So dogs that had no copies of chondrodystrophy, dogs that had 1 copy of chondrodystrophy, and dogs that had 2 copies of chondrodystrophy. And what you can see here is that the dogs that have no copies of chondrodystrophy also do not have any mineralized discs. Dogs with 1 copy had on average 4 mineralized discs, and dogs with 2 copies had on average 10 mineralized discs. So this is consistent with the studies done in standard dachshunds that showed that the chromosome 12 locus had an additive effect on disc calcification, and it's also consistent with our study on the dogs that had actually had disc surgery, that there's an additive effect on disc mineralization. Interestingly, these 3 dogs that have the little stars on their dots, those are dogs that had herniated a disc, but they were completely clinically normal. So at some time in their lives, they had disc herniation, but that herniation was subclinical. It didn't hit the spinal cord and cause paralysis, so the owners didn't notice that there was anything going on with those dogs, and yet they had actually already herniated. So again, this sort of points out that just because a dog hasn't become paralyzed from this mutation doesn't mean that the discs aren't abnormal or that there's not something going on with them. So chondrodystrophy has shown signs of really strong selection over the years, and this is an example of a painting that I found that was done in 1480. And you can see that there's clearly a spaniel-like dog there with kind of short legs and long ears, and I expect that this dog probably had chondrodystrophy. So how do we study the other effects of chondrodystrophy and chondrodysplasia? We know that they are under selection by breeders. Breeders like them, and people liked them a long time before any of these breeds existed. Otherwise, they wouldn't have kept them around. So what we needed was a breed that segregated chondrodystrophy and chondrodysplasia. So we wanted a breed that had normal copies of both of those genes, or both of those areas, plus the retrogenes. And I identified such a breed in Switzerland. It's called the Schweizer Niederlaufhund. It was conveniently in Switzerland, so I got to go to Switzerland and live there for a year to do this project. So this breed is selected purely on the fact that they have really good noses and they're really good hunting dogs. And they don't care so much about how long their legs are, although they are small Swiss hounds. So you can see in the pictures here that some of them are taller than others. And we measured these Swiss hounds, took lots of accurate measurements with calipers. We also just got to play with the adorable puppies. And then we genotyped those to see what the effects were of these different FGF4 retrogenes on different parts of the body. So those results are summarized here, and this slide is a little bit complicated. The orange dots represent the chondrodystrophy retrogenes, and the darker orange means they have 2 copies, and the lighter orange means that they have 1 copy. And the blue dots represent the chondrodysplasia retrogene, and again, lighter blue is 1 copy, and darker blue is 2 copies. And the little gray dots mean that there was no statistical significance. So first I want to tell you that there was no significant effect on chest depth or width. There's a lot of breeders that had told me that they thought that there might be based on what they were seeing when they bred dogs that did and didn't have chondrodystrophy, but there wasn't. There was an interesting effect that we didn't necessarily expect on the width of the skull. So chondrodystrophy made the skull wider in these dogs, and it was statistically significant, and it made them 20% wider, which is actually even more than you get with a sex difference, so males having wider skulls than females. So the percentage change is shown on the bottom. So the dots that are to the left of the little dotted line means that they decreased in percentage, and the dots that are on the right means they increased in percentage. So height definitely decreased additively, so meaning that the more copies of FGF4 you have, the shorter you are. And in particular, the height due to the antebrachium, which is the forelimb of the dog, the radius and ulna, was where the biggest effect was. So chondrodysplasia shortened that. 2 copies of chondrodysplasia shortened that by a significant amount. And all of the ones that are colored are significant. The one thing that isn't on here is valgus, and that's the thing that we started this whole story with in Nova Scotia duck-tailing retrievers, that their legs turn out. We still don't know why Nova Scotia duck-tailing retrievers have this phenotype, because it's independent of chondrodystrophy. So in other breeds, in the Niederlaufhund, it's all chondrodysplasia that causes valgus, and chondrodystrophy absolutely doesn't. So we still don't know why some of the tollers have crooked legs. So the last thing I want to tell you about is the ears. So there was a study done by Ostrander's group that showed that the ear tips are wider in some breeds of dogs, and that seems to map to the same region of the chromosome. They're not so sure it's chondrodystrophy. I think it probably is, but definitely the chondrodystrophic breeds have wider ear phenotypes. So this is a summary of the phenotypes. So chondrodystrophy causes intervertebral disc degeneration. It increases the risk of disc mineralization and increases the risk of disc herniation. And it decreases the age of disc herniation by 2 to 2.5 years. These are shown in red because they're negative effects. Sort of positive effects are things that maybe people like, or that it shortens the legs a little bit. Not as much as chondrodysplasia, but still a little bit. It increases the skull width, and it may increase the ear width at the tip. So this is why people may have selected for it. From a counseling standpoint, there are breeds of dogs that are homozygous, meaning every beagle that we've tested has 2 copies of chondrodystrophy. French bulldogs and the spaniels all seem to have 2 copies, or a really high number. So in those breeds, there isn't anything that can be done to lower the level of chondrodystrophy. And that's true in the miniature dachshund as well, although the standard dachshund has some variability. But the breeds that have less of this red bar and a lower allele frequency of chondrodystrophy, those breeds could select against it and reduce the amount of disc herniation. I think that the number of surgeries that are going on around the world is a compelling reason to try and get rid of this mutation. And if people still want to have short-legged dogs, then the chondrodysplasia locus actually makes dogs' legs much shorter. So breeders are also worried about how this might affect diversity. And this is from a paper we recently published looking at the effect of inbreeding on body size and morphology and health. And it's a little bit hard to understand, but the inbreeding coefficient is shown here on the left, and this is the average inbreeding across a large group of breeds, 227 dog breeds, where we found that the average inbreeding coefficient was 0.25, which is really, really high. So I'm not sure that selecting against chondrodystrophy is going to make this a whole lot worse than it is in purebred dog breeds. Certainly if it's done slowly over time, it shouldn't. So in the next slide, I just want to acknowledge the work that I've talked about today, which is from a ton of different people in the lab, a couple of graduate students, and some fantastic collaborators. And I would be happy to take any questions if you have them now.
Welcome, everyone. We are now live with Dr. Danika Bannasch. Thank you very much for that informative presentation.
Oh, you're welcome. I was happy to do it.
Yeah, so I know this is a topic that a lot of people care about quite a bit, from breeders and veterinarians to certainly everyday dog owners. So I think we're going to have lots of questions on this. As a reminder to our viewers, please feel free to continue submitting questions. We have a few that have already come in. We'll answer as many of them as we can as we go forward here. And just as a scheduling note as well, the closing talk at 4:30 p.m. Eastern in about 4 minutes has been pushed back 15 minutes. So that will start at 4:45 p.m. Eastern in 19 minutes from now. So we'll make sure to end this Q&A session in time for that talk. As for this though, let's jump into it. And just to start off, Dr. Bannasch, what would you say kind of led you to this field in the first place and sparked your passion for this topic?
Yeah, so I'm a little bit unusual in that I've always wanted to be a dog geneticist, like literally since I was in junior high school. So this is really my dream job. I love dogs. I'm a dog breeder, and I show my dogs and exhibit my dogs, and I love genetics, and it's great to be able to do both of those things. So.
Terrific. Well, let's get into the questions here. There are a few related to activity. So the first one, I am a breeder of Portuguese water dogs. I recently lost my 2-year-old bitch to this disease following a ruptured disc that paralyzed her. Before this, I'd never heard of CDVY, IVDD. So I think there's probably a couple of questions here you could get into. The first, are you aware of specific events or actions that cause this to express itself?
Yeah, so we always have a kind of a similar history that, you know, the dog maybe was running around in the backyard, maybe playing, but not really doing anything extreme. And then the result is this sort of traumatic injury from the owner's perception. And that's unfortunately what happens with this disease. So as you heard in my talk, if the dog has this mutation, all of their discs are abnormal. And they're abnormal from a really young age. We sort of only seem to notice that there's something wrong when they have this catastrophic herniation event. But they were abnormal before that, and we don't entirely understand what makes this happen or why it happens. There is, like I said, sometimes a little bit of physical activity involved, but you know, nothing really horrible or crazy. That being said, you can attempt to prevent it by keeping dogs in really good shape and good body condition and lean. And there's been studies in dachshunds that said that dachshunds that had to go up and down stairs every day, whose owners lived on the second floor, were less likely to herniate, for example. So you'd sort of think, well, stairs would be a bad idea for dachshunds, but it turns out maybe that those dogs were in better shape. So.
Got it. So then the I think related part, you know, there was another question that came in about, you know, dock diving, weave pulls, jumping. You know, there's some common activities that people are concerned about. Do any of those stand out as particularly problematic?
Yeah, I mean, like I said, you know, it's probably better that they're in shape than if they're not in shape, but some of those certainly make you nervous. That being said, there's a lot of dogs that do agility and dock diving and don't ever have a herniation event. I'm probably not necessarily the person to ask about that part of it because I'm the geneticist, but I think it's more important to keep dogs in shape. So.
Okay, so tapping into the geneticist side, there was a few questions. I think each of the people that submitted those topics also asked about breeding.
Right.
So one says, what breeding recommendations would you make in these breeds, specifically the Portuguese water dog? And then I think just in general, any other kind of guidance or advice you have about breeding decisions related to IVDD, CDDY.
Yeah. So, I mean, this mutation is a real challenge for dog breeders because dogs that have the mutation can be completely asymptomatic and normal, and yet we're saying, look, this is a dominant disease and a problem, and they can pass it on, and these uncommon events where they herniate are the ones that we get really upset about. So from my standpoint, it's using prevention, right? We don't want a dog to herniate, and they're much less likely to herniate if they don't have the mutation. So it's something where you're sort of thinking long term, like how can I help the health of my breed? And in Portuguese water dogs, where the allele frequency isn't that high, you can select really strongly against it, and it's not going to be a problem. There's plenty of Portuguese water dogs that have the normal version of the chromosome that can be used for breeding. So that might be a breed where I would select more strongly. I think someone else asked about Cavaliers, which, as far as we've seen, always are homozygous and have 2 copies of the mutation. Someone says, why are they not presenting with disc issues? They are presenting with disc issues. They definitely have disc disease and herniate just like other breeds. I think Cavaliers have a lot of other health problems that maybe are more in people's sort of forefront. But so in a breed like that where they all have it, obviously you can't breed it out without crossbreeding. So there's a big difference in counseling between dogs that have a lower allele frequency, where there's plenty of normal dogs to breed, and dogs where there aren't any normal dogs to use for breeding.
Got it. Yeah, and that is a topic we certainly encounter quite a bit at Embark as well. Just questions from breeders about, based on this result in my particular breed, how should I, factor that into my breeding decisions? So on I think maybe the other side of the spectrum, there's a question about larger dog breeds. Someone asks about, what about IVDD in larger dogs like Rottweilers? Obviously, they don't have the gene for short legs, but do they have the other marker from this paper?
Sorry, the question scrolled by and I missed that one.
Right.
So as far as I'm aware, we haven't found chondrodystrophy in Rottweilers. I would just caution everyone that chondrodystrophy causes very mild leg shortening. Like it's not very obvious to many people that their dog has slightly shorter legs. So I wouldn't really think about it only in dogs with short legs. Australian Shepherds, for example, and Portuguese water dogs are not, Chesapeake Bay Retrievers are not dogs with short legs, and yet they all can have chondrodystrophy, although I don't think that Rottweilers do. So there are other ways that a dog can have disc herniation or a back problem. There's lots of different ways in people as well. So it's possible for dogs to have disc disease that don't have chondrodystrophy.
So we were kind of chatting backstage a little bit about, I think, the sort of third version of the question, which is for mixed breed dogs. And, the mixed breed dog, at least visually, may not appear to be a breed that is typically susceptible to IVDD. But it may or may not test as at increased risk genetically. Can you speak to that kind of topic at all?
Yeah, like I said, I mean, the phenotype can be a little bit subtle, and certainly in a mixed breed dog, it could be really difficult. Does it have a wide head compared to what? And does it have rounded ears compared to what? Because you don't really necessarily have other dogs to look at and the shortened legs. In one of the studies that we published where we looked at dogs that had intervertebral disc herniation in the hospital, there were a number of mixed breed dogs that were in that category, and they actually seemed more likely to herniate than some of the purebreds. And I don't know if that was a sample size issue or not, but it's kind of interesting that it's possible that breeds that have 2 copies of the mutation that are homozygous for it, or some breeds, may have been able to select against early herniation, because if they herniate and they're paralyzed, then they're probably not going to be used for breeding, and maybe in the mixed breed population that's gone away.
Got it. So a few other questions that have come in here. Not sure the extent to which you can speak to them, but the first says, I recently produced a litter with 4 pups with short legs, one with crooked front legs, and also the outside toe on one is not in the normal place. These are Welsh Springer Spaniels, so a litter of 8 and 4 were normal. So what test do you recommend?
Well, so remember in the talk that chondrodystrophy doesn't actually cause valgus or crooked front legs. That's normally caused by chondrodysplasia. And it's also can occur without either of those 2 mutations. So I don't know that that either of those tests might explain what's going on in the Welsh Springer Spaniels, but they could. It's possible that chondrodystrophy could be segregating there in that breed. They're relatively rare breeds, so we haven't tested a lot of them, so I don't know what the allele frequency is in the breed.
Got it. So I see a question here that might have to do with kind of the rate at which dogs might test as at increased risk genetically versus the penetrance. But so a question here says, you indicated single carriers had an average of 4 discs calcified and double carriers had 8. Were there any carriers that had no discs calcified?
Yes, I think this is referring to the CT study that we did in Nova Scotia duck-toling retrievers. We had 8 dogs that had 1 copy of chondrodystrophy, and one of those did not have calcified discs at the time that we did the CT scan, but that dog was only a 1.5 years old. So it's certainly possible that it could have developed calcified discs later. All of the dogs with 2 copies had calcified discs, and none of the dogs without chondrodystrophy had calcified discs. They were roughly around the same age, but the one dog was younger.
Got it. There's a question about how common this allele is in Portuguese water dogs. I know at least from Embark's perspective, this is information we can provide from our own testing in terms of dogs tested by Embark. Here's the rate at which the allele appears, which again is different from how often does this, do dogs show clinical signs appearing. So I don't know if you could speak to either of those topics. This asks about Portuguese water dogs again.
Yeah, so I don't know what the allele frequency is overall in the population in a large sample size. If I had to remember, because of course I have the allele frequencies memorized for all 300 breeds. I want to say it's about 20%, but I could be wrong. And Embark probably has more Portuguese water dogs tested. And your other question was what percentage of those would have disease associated with it, or? Yeah,
I referenced penetrance, but I mean, again, we were speaking earlier about there are breeds that have a very high rate of the allele appearing and breeds that have a very low rate appearing, and then the rate of penetrance or clinical signs might be different across those.
Yeah, and I mean, I'm not even sure if I would call it penetrance, because we believe, and there's only been 7 or 8 breeds tested now, but we recently tested Nova Scotia duck-toling retrievers. The discs are abnormal. So it's 100% penetrance as far as disc abnormality. It's just, and we even think that a lot more of them are herniating and they just aren't showing clinical signs because the disc herniation didn't occur dorsally so that it slammed into the spinal cord. It just went to the side, which maybe caused back pain, but not enough that anyone noticed to take the dog to the veterinarian. So I think you can talk about the penetrance of disc extrusion or disc herniation, and that's relatively low, but they all have abnormal discs, and I suspect that those abnormal discs also cause them problems, but we can't ask them whether they have back pain.
Right.
I do. Anyway.
So let's see, we've got time for just a couple more questions. One here asks about genetic recombination, and does that play a role in expression of the disease? And then it goes on to ask about, should carriers be phased out over time from breeding programs?
There's a lot of Portuguese water dog questions.
Yes.
So technically they're not carriers because it's dominant, so they're heterozygotes. But yes, I mean, if you want to reduce the risk of disease, then yes, over time you should breed away from it. And, I kind of recommend in a breed where the allele frequency isn't very high that you do a breed and replace strategy. So you breed the dogs that are heterozygous, test the puppies, and pick dogs that don't have it to go on with. You also have to think about explaining to the owners of those dogs that have the mutation what the risks are and making them aware of what could happen with their dogs when you're selling them. And I think that that's a challenge, right? It's a challenge for breeders to understand it, and it's a challenge for owners to understand it as well, that, they're getting a puppy that has a risk of disc herniation.
Got it. So as a final item, or I should say a final question for you, is there, kind of given our audience of breeders, veterinarians, researchers, and others, I think just passionate about canine health, do you have a call to action or a key message that you want to leave everyone with?
Yeah, we were talking about that earlier, and I think the thing to me, and I've gotten a lot of flack from breeders for this test, and people that were disappointed that not every dog that has the mutation actually herniates. And it was really frustrating for me because as a veterinarian, this is the single most painful disease in dogs. And when I got frustrated, I would go to our neurology ward. Every single day at UC Davis, we do surgery for this disease. Every single day there's a dog in excruciating pain at this hospital, let alone around the world. So it's the most painful condition, and there's a way that we could reduce that overall. So people get really wrapped up in their own breed, but for me, it's about trying to relieve pain and suffering for dogs.
Got it. Terrific. Well, thank you again very much for your participation here at the Canine Health Summit, your fantastic talk, and now all of your time spent answering questions. So we really appreciate it.
You're welcome.
So for all of our viewers and attendees, if you switch over in just a couple of minutes to our closing session, we'll recap the last 2 days as well as share some details about where to find the recordings and other ways to engage with us going forward. I also wanted to do just a very quick call out to some of our Embark team members, in particular, Mackenzie Pearson and Sarah Giblin, who have been incredible behind the scenes, Maddie Lies, and many others who have answered chat throughout the booth and the conference, to Karen Hinchy, who has been a fellow Q&A moderator, and to Ryan Boyko and Dr. Adam Boyko for their kickoff sessions and involvement in many ways. And that leaves out probably 15 or 20 other embarkers who have been involved in this work across the last 2 days and the last 6 months or more preparing for it. So thank you very much to everyone who has been involved, and we'll see you over at the closing remarks very shortly.